The hyaluronidases are a family of enzymes that degrade hyaluronic acid. Karl Meyer classified these enzymes in 1971 into 3 distinct groups, a scheme based on the enzyme reaction products. The 3 main types of hyaluronidases are 2 classes of eukaryotic endoglycosidase hydrolases and a prokaryotic lyase-type of glycosidase. In humans, there are 6 associated genes, including HYAL1, HYAL2, HYAL3, and PH20/SPAM1. Use as a drug, by catalyzing the hydrolasis of hyaluronin, a constituent of the extracellular matrix. Hyaluronidase lowers the viscosity of hyaluronin, thereby increasing tissue permeability. It is, therefore, used in medicine in conjunction with other drugs to speed their dispersion and delivery. Common applications are ophthalmic surgery, in combination with local anesthetics. It also increases the absorption rate of parenteral fluids given by hypodermolysis, and is an adjunct in subcutaneous urography for improving resorption of radiopaque agents. Hyaluronidase is also used for extravasation of hyperosmolar solutions. Brand names of animal-derived hyaluronidase include Hydrolases, which has been FDA approved as a thimerosal-free animal-derived hyaluronidase, Vitrase, Amphidase, and Widus. Widus, however, is no longer manufactured. On December 2, 2005, the FDA approved a synthetic human hyaluronidase, Hyaluronic. Role in cancer. The role of hyaluronidases in cancer is controversial. Limited data support a role of lysosomal hyaluronidases in metastasis, while other data support a role in tumor suppression. Other studies suggest no contribution or effects independent of enzyme activity. Non-specific inhibitors or crude enzyme extracts have been used to test most hypotheses, making data difficult to interpret. It has been hypothesized that, by helping degrade the ECM surrounding the tumor, hyaluronidases help cancer cells escape from primary tumor masses. However, studies show that removal of hyaluronin from tumors prevents tumor invasion. Hyaluronidases are also thought to play a role in the process of angiogenesis, although most hyaluronidase preparations are contaminated with large amounts of angiogenic growth factors. As previously mentioned, there are 6 hyaluronidase genes in the human genome, 3 of which can express active hyaluronidases. Role in pathogenesis. Some bacteria, such as Staphylococcus aureus, Streptococcus pyogenes, and Clostridium perfringens, produce hyaluronidase as a means of using hyaluronin as a carbon source. It is often speculated that Streptococcus and Staphylococcus pathogens use hyaluronidase as a virulence factor to destroy the polysaccharide that holds animal cells together, making it easier for the pathogen to spread through the tissues of the host organism, but no valid experimental data are available to support this hypothesis. Role in fertilization. In most mammalian fertilization, hyaluronidase is released by the acrosome of the sperm cell after it has reached the oocyte, by digesting hyaluronin in the corona radiata, thus enabling conception. Gene-targeting studies show that hyaluronidases such as penthouse 20 are not essential for fertilization, although exogenous hyaluronidases can disrupt the cumulus matrix. The majority of mammalian ova are covered in a layer of granulosa cells intertwined in an ECM that contains a high concentration of hyaluronin. When a capacitated sperm reaches the ovum, it is able to penetrate this layer with the assistance of hyaluronidase enzymes present on the surface of the sperm. Once this occurs, the sperm is capable of binding with the zona pellucida, and the acrosome reaction can occur. References. External links: Hyaluronidase at the U.S. National Library of Medicine. Medical subject headings.